Welcome you back, my dear students. So today we are going to discuss about a very important topic. I have already discussed, I think, in the class. It is nothing but the molecular marker, especially the molecular DNA marker. Okay. So here are some features of the molecular DNA marker. First is the DNA sequence, molecular DNA marker. What is molecular DNA marker actually? A DNA sequence that is readily detected and whose inheritance can be easily monitored. Okay, so this is a marker means a particular known sequence which can detect a particular gene or a particular sequence of the whole genome. The uses of molecular markers are based on the naturally occurring polymorphism, that to detect the polymorphism among the populations or among the various type of populations or genetic diversity, or to check the genetic distances. You can use the molecular marker. Okay, a marker is a gene of known function, that's already told, and location that allow the studying the inheritance of the gene. Okay, so marker is always a known sequence, is a gene, maybe a gene or may not be a gene. Okay, all the markers are not always the gene. Okay, which allow the studying of inheritance of the gene. Actually, it helps to study about the inheritance pattern of a particular gene from generation to generation. The marker must be a polymorphic, that is, it must exist in different forms so that chromosomes carrying mutant gene can be distinguished from the chromosome which is normally gene by a marker. So polymorphic, the marker must be a polymorphic. Polymorphic means having ability to detect different types of genome. Means in a whole genome, wherever the see in the whole genome there are different nucleus sequences. So that means in the whole sequences, if the markers are able to detect the whole sequence, so it is a very polymorphic marker. And based on this polymorphism, polymorphic information content is also calculated. Okay, so polymorphism involves existence of different forms of some gene of a population. So say here are some example: RFLP, restriction fragment length polymorphism, RAPD marker, AFLP, SSR, SNP. So we will discuss some important examples here. So first is the RFLP. RFLP is the full name of restriction fragment length polymorphism. Okay, so RFLP is the organism can be differentiated by analysis pattern derived from the cleavage of their DNA. So RFLP means restriction fragment. Restriction enzymes is required which can cut the DNA, and based on the DNA cut, DNA fragment will produce. Based on the sizes of the DNA fragment, the polymorphism will be detected. Okay. So the RFLP restriction enzyme digested DNA is resolved in gel electrophoresis. You can use the agarose gel or even polyacrylamide gel, PET gel. Generally, we use for protein fingerprinting. So you can check. Okay. So specific binding patterns can be visualized by hybridization with level probes. So here in the restriction enzymes, you can attach some specific radio level probe which can actually detect the target DNA or function. Means using the restriction enzymes, along with that you can attach some radio level probe. That probe like will work like a spy and will ultimately attach to your target sequences. So based on that protocol you can identify your target gene. Okay, next is the advantage is high reproducibility. So codominant allele, it is very important thing, and detect coupling phase of DNA, when the overlapping genes are there, overlapping sequences are there. Reliable markers in linkage and breeding analysis. In case of crop breeding, RFLP is utilized, and easily determine the link trait present in both homozygous and heterozygous. This is also very important point. And these advantages are require large quantities and high molecular weight DNA. It need the high molecular weight DNA means a good amount of DNA. With a small amount of DNA, small size of DNA, you cannot work. Expensive process means as the restriction enzymes and radiolabel probes are very costly, time consuming also, and labor intensive. Next is the application. Next, okay. So application is the phylogenetic studies, gene mapping. Phylogenetic studies mean in a population genetic diversity study, phylogenetic study. Okay, the evaluation, how a plant species, how an animal species evaluated. This is comes under the phylogenetic study. Gene mapping is also important. Gene mapping means suppose you are working on the genome, is a very funny term, working on the genome. That means you are reading all the nucleotide sequences of the particular whole genome. So based on the genome, you are just mapping the good gene, bad gene, or some based on their physiological activities, based on their morphological, based on their biochemical activities. You are just mapping the gene based on their group and growth and developments. So there are certain domains of functions or activities or trait. So accordingly, you are mapping the gene in which portion is at which part all the genes are present responsible for growth, and where the genes are present responsible for biochemical functions. So likewise, based on the function, you can map the gene. And DNA fingerprinting also is very important thing. DNA fingerprinting means the detection of polymorphism and studies of gene flow. Gene flow means how gene is inherited from one generation to the next generation. This is called the gene flow. So gene flow is a very important point. So this is the application part of the RFLP. So I once again I repeat you, the RFLP is a very important thing. That's why, because there are lots of applications, phylogenetic DNA fingerprints, studies of gene flow, gene mapping. Here, see, you can see the disadvantage is mainly the labor intensive, time consuming, expensive process means all the chemicals and reagents are very costly, and that is the thing. Otherwise there are more advantages. Is highly reproducibility, is means the having the polymorphism detection ability is high. Okay, so codominant allele is also is a very important thing. Codominant allele means suppose if, on a particular gene, on a particular chromosomes, a gene, two genes or three genes are simultaneously showing the dominant nature. So in that particular case, which gene is actually properly working over which one? So that study can be done using RFLP. Okay, so and coupling is also the joining of another DNA sequence or coupling phase of DNA. Okay, so in breeding also is very important. Crop breeding RFLP analysis is done for marker assisted breeding or marker assisted selection is also easily determine the link trait present in both homozygous and heterozygous is very important in terms of breeding aspect. Okay, so I hope in this video you will enjoy, you have already enjoyed this RFLP technique. We'll later discuss the next molecular DNA marker. Thank you for watching.